So, good evening, class. Good evening, class. Good evening. Limo, can you hear me? Can you hear me? Mr. Limo, what can you see from the screen? Yes, what can you see?
Yes.
You can see something from the screen. Good evening. Let me call the register to know those who are present. Priscilla Warimu. Priscilla Warimu. Limo Koskei. Dismas Cabernet. Dismas Cabernet.
Present.
Yeah, that is good. Huh?
Present.
That is good. Then Joan Coril. Eunice Sawe. So we will start our lesson of today. We will start our lesson of today. And today we are going to learn our topic today in the applied biology, that is biochemistry. It is one of the units that is taught to students that are taking applied biology course program. And we are going to look at the biochemistry as a unit, and under it we are going to study what we call proteins. Huh? Are we together? Are we together? So we are going to look at the biochemistry of proteins. What we are going to study today, our objectives are going to be, 1, we are going to define proteins. Yes, we are going to define proteins. That is our first objective. Then we will also look at the types of proteins. Then we will also look at, we are going to state classes of amino acid. Then describe the basic structure of amino acid, as well as also the 5th objective, that is discussing the methods of separating amino acids. Are we together? Class, are we together? We'll start from looking at the second objective, the first objective, that is the definition of proteins. Because at the end you should be able to define what a protein is. You cannot talk about proteins if you don't know the meaning of it. So protein, as we said, just like other lipids, carbohydrates, we also have what we call proteins, which are also large complex molecules that play primary important roles in our body. They are critical to most of our work done by the cells, and are required for structure and functions and regulation of the body's tissues and organs. In class 5, that is in primary science, you used to talk over some of the micromolecules that we have that are of very importance, that is of biomolecules. Those are the biomolecules that are useful to our life. One of them is proteins, and we said proteins also help in repair of our tissues, those of dead tissues. So we also have these ones as one of the macromolecules in the biochemistry of cells and of a living organism. Then we'll move to the types of proteins. We'll move to types of proteins. That is, what are some of the types of proteins that you know? So these ones, majorly we are classifying these proteins based on the functions that they perform in our cells. So one of them, we have the antibodies. You are aware of the antibodies. You are aware of the antibodies, and you know the roles of the antibodies. Some of them are proteins. Then we also have the contractile proteins. That is, we have contractile proteins in our body cells. Then we also have other proteins, that is enzymes. We can also have enzymes. Enzymes, majorly they are proteins, and that is why when you are doing your studies in biology, that is in Form 1, we are stating some of the properties of enzyme. We are saying that they are denatured. So that is making them to be what? Some of the examples of the proteins. Proteins are always denatured by temperatures. So we have that. Then under low temperature, they become inactive. So enzymes are also having those features of proteins, just like other proteins. Then we have hormonal proteins. Then we also have structural proteins. Like in our cell membrane, we always have a type of proteins that is combined together with the carbohydrates plus these proteins. Then we have what we call the glycoproteins. That is a protein that is made up of carbohydrates. Then we have storage proteins. We also have storage proteins and transport proteins. So this one we classify the proteins based on the functions that they perform in our body cells and tissues. Let me see if we can have someone who want to join and is in lobby. That is good. So that is what is made as per the functions classification, the functional classification of proteins. I also want to talk about the amino acids. Amino acids are very important because they are the building block. These ones are the building blocks of the proteins. This is the building block of our proteins. Just like we talk the last time we were talking about the carbohydrates, and we were talking of the building block of most of the carbohydrates. And we said that carbohydrates, they have a building block, that is the monosaccharides. So that when we have two of them being joined together by what we call the glycosidic bond, we were having what we call the disaccharides. But when we have now 3 to 10 chain of the monosaccharides, we are having what we call the oligosaccharides in carbohydrates, and we are also talking of the polysaccharides. That is now more than 10 units of sugars. When you bring them together, join together with the glycosidic bonds, we could have the polysaccharides. But today, just like the carbohydrates, you can also have several amino acids joined together by a bond that we are calling the peptide bonds. So we have a bond here, the bond that is joining amino acid plus another amino acid, so that we produce maybe dipeptide bonds or tripeptide bonds, tetrapeptide bonds, and there are very many based on the number of the amino acids. So we have some of them here. We can classify them based on the reactions of water. So we have the non-polar and neutral amino acids. We also have the polar and neutral amino acid. Then we also have acidic and polar amino acid. Then we can also have the basic and polar amino acid. So we will talk on the structures. We have an objective that is talking about the description on the structures of amino acid. We are going to look at it there in the next objective. I also want to talk about the structure. Very good. The structure of amino acid. The structure of amino acid. How is amino acid? What are these of the elements that make it amino acid? Some of these elements, we are going to look at them. So we have a chain there. We have a chain there. That is let me do something here. Kidogo. There is a chain here that I want to talk about. That is amino acid. There's something I've done. So in amino acid, that slide is not meant for that. In amino acid, this is how we make amino acid. In amino acid, we always have amine group. That is amine group. You know how to write the amine group. That is NH. There is a typing error that occurred in that my slide, but we always know that amino acid is made up of the carboxylic group and hydrogen chain. Then we have R group. This R group, the R group is normally varying depending on the number of or the various type of or the various types of elements that is combined to form that hard group. And we also have the carboxylic group that is also there, and a nitrogen atom. So that is what we have as the structure of the amino acid. I'll send you the real diagram that is showing the structure of the amino acid. Now I want to go to what is next now, methods of separating amino acid. How can we separate amino acids? You can have a mixture of amino acid, several of them. And I don't know if I may ask, have you had some of the examples of amino acid? Can you name any amino acid that you know? Can you name some of the examples of amino acid that you know? Anybody to name any example of amino acid that he knows or she knows? From the class? Sawi. Eunice Sawi. Dismas. Dismas.
I'm not sure.
You're not sure. Limo.
Yeah.
Limo. Eunice. Sawe. So if you don't know it, I will tell you some of the examples of the amino acids that we have. So we can have the thymines, those ones that you are doing in the DNA structures. We are talking of the gene structures, the genetic structures. Most of them are examples of the amino acid that are there. We also have even guanine. The guanine is also an example of the amino acid that you can talk of. So we have very many of them. We have up to 20 amino acids that you should be knowing. Know how to draw their structures and how they look like. We have very, very many examples of the amino acids. So at times they come and mix themselves, and there is also a time that you may want to separate them, and therefore that is why I'm teaching you on this objective of how you can separate these amino acids. So one of these methods that you can use to separate the amino acid is the paper chromatography. The paper chromatography. We can separate the amino acid using the paper chromatography. That is, paper chromatography can separate and identify the combination of their known amino acid. Spraying with ninhedrin. So this ninhedrin, when you apply it in the spot where we have those mixtures, it will be able to separate them into several distances so that you can see each of them. That is how it reacts strongly with the colored product. So can be used to determine the location of the amino acid in the chromatogram. That is purple in color. So we are moving. There is someone who wants to join our class. Let us let him or her in. We will let him join as we continue. So we have, that is one of the methods that we can use to separate the amino acid. Then we can also separate the amino acid by using ion exchange. So the amino acids can be separated by elution with sodium citrate. So this is the chemical substance that we use as a buffer, and they are displayed as functions of the pI values by the changing of the pH of the buffer solution. The effluent of this column is collected and analyzed for the amino acid content. So there is something you are seeing there. Can you see that abbreviation pI? Class, can you see that? Dismas?
Yes.
Priscilla?
Yes.
You can see that?
Yes.
So PI, do you know the meaning of PI? Huh?
What do you think?
Can someone try guess what do it mean by the PI when we are talking of the separation of amino acid using the ion exchange? You don't know it.
Can
So,
I try?
huh? You want to try?
Can I try?
Yes, you can try.
Yes.
Try it.
Phosphate iodine.
Huh?
Phosphate iodine.
That is a
Phosphate iodine.
that is a good trial. That is a good trial. You are, I can see you are a good student. At least you can try. Huh? You have to try. So what it means by this PI, in fact, in your back in the biochemistry of the proteins, you used to talk of something we are calling the isoelectric point. Huh? So here we have used PI, point of the isoelectric. Huh? That is how electricity always influence the movement of this amino acid, this different amino acid. So PI is simply point of isoelectric. Then or you also can change it, high P ama PI. The high P, that is the isoelectric point or point of isoelectric. That is good. Iso simply means the same. The same. So that is what it entails by the separation of amino acid by ion exchange. We will also look at the next slide. That is our slide there. I want someone to read what you can see in that slide that I've shown in the screen. Can someone read whatever you can see in the screen? Yes? Can you see?
Electrophoresis of amino acids. Introduction. Electrophoresis is a separation technique based on the movement of charged atoms under the influence of an electrical field. This technique is primarily used for the separation of amino acids and peptides on the basis of their charge.
That is very good, Priscilla. So in electrophoresis of amino acid, this one, we create what we call, we put these amino acids in what we call an electrical field. So in that field, there is how different amino acids react based on the electrical difference charge. So that is, you can be able, because they have different reaction to this electricity, you will be able to see those mixtures, the content that were there in that mixture of the amino acid. So that is what we have there. You use electricity. Simply, this one, you involve the use of electricity. Are we together? Are we together? Yes. So we'll also look another one, that is the separation of amino acid by a thin, that is by a thin layer of chromatography, a thin layer of chromatography. So I want someone, Dismas, can Dismas see the slide?
Yes.
I want you to take us through what you can see from the slide.
Separation of amino acids by thin layer chromatography.
Mm.
In the simplest form of the technique, a narrow zone or spot of the sample mixture to be separated is applied near one end of the TLC
So that TLC, what does it mean? That abbreviation TLC?
plate and allow to dry.
Continue.
Thin layer chromatography.
Thin layer chromatography.
Very good. Thin layer chromatography. That is the abbreviation. Continue.
So the strip or plate is then placed with its end dipping into the solvent mixture, taking care that the sample spot stroke zone is not immersed in the solvent.
Very good. So that is what it entails by the separation of amino acid by what we call the thin layer chromatography. So we have said it's just a simple technique, the simplest technique, because you simply apply a zone, you create a zone on the spot of the sample mixture. Because I said the reason why we are separating this amino acid, at times they can be found mixing with other amino acid. So when you find it that, you just apply what? To the end. To the end of that chromatography paper, just a thin layer. That then the strip or the plate is then placed with this end dripping to the solvent mixture, then taking care of the sample spots, and it is not immersed in a salt, not immersed. So that is what it entails by the separation of a thin layer chromatography. I will take you to the lab and we will do this. They are very enjoyable when you do them practically. This is a practical topic. It is very enjoyable when you can do it in the lab. Very enjoyable topic, and you will like it. I know theoretically it may be difficult, but when you see it, it will be good. There is something I want to give you. There is something I want to give you now that we are talking about the proteins. Now that we are talking about the proteins, what are some of the sources of proteins that you can have? Can you give me some of the sources of proteins that we have?
From eggs.
Very good, from egg. Dismas, from the mess. You always go for lunch in the mess. What do you normally eat there in the mess? That is source of protein. Because our mess, the balanced diet should also focus on providing these proteins. Proteins is very important micromolecule in our body, so we cannot ignore it in our balanced diet. Dismas?
Meat.
Very good. You always eat meat. Maybe you go for ugali special, then you will find some pieces of meat there. That pieces of meat is source of protein. Egg also, some of you in the morning, you always go there. Boil and egg, boil egg. You know, that egg is a source of protein. Very good. Others who is in the class and want to try another source of protein? Who again want to try? Huh?
Beans.
Beans. Very good. Beans. Can we clap for Priscilla? Priscilla has been an active member in our class, and he has answered, make some trial, also giving some correct answers. Can we give some clap? Very good. That is good for clapping for your fellow. Priscilla, Unisawe. Is Unisawe in? So maybe our network is not making it to be with us in the class, but I know it's with us in the class because I can see the face. That is good. So we have some of the examples of protein sources of proteins that we have. We have healthy protein sources include the eggs, that one you have been eating. We also have the nuts, those who like ground nuts, and you what the nuts family. So those ones are sources of the proteins. Then we have lean meat, just meat. Meats are always sources of proteins. Then we have fish. Fish is also a source of protein that we have. Then we have dairy product. Maybe you can have that cow, slaughter it, and then you eat it as a meat, or you go for the milk product. Those ones are also examples of proteins that we have. Then we have certain grains. Some of the certain grains can also contain these proteins. They can also contain these proteins. So today we have learned. We have learned. And just to know whatever you have learned today about proteins, Dismas Cabernet, Joan Corel. Do we have Joan Corel? Joan? Sawe? Is Sawe in? Priscilla? Priscilla?
James.
Yes? Yes, I can see you are in. What have you learned today about proteins? Joan?
I've learned their sources.
You have learned?
I've learned sources of proteins from meat, eggs, milk. Then I've learned about the abbreviation TLC. Then I've learned about the meaning of PI. Yeah.
So at least you have learned some things, didn't you? So I don't want to leave you without giving you some assignment. Are we together? I want to give you this assignment, and this assignment you will collect it in the next class. You'll collect it in the next class. You will collect it in the next class. Are you seeing the screen? Priscilla, are you seeing the screen?
Yes.
I want you to read whatever is written in the screen after I've written it there. Are we together?
Yes.
I want you to read for them this so that they can do it. So this is the assignment that I want you to do today, and I will collect it in the next class. Failure to do it, you will not be in my class, and I will take you out. Are we together? So Priscilla, can you read for them the assignment?
Describe functions of proteins in the body, 20 marks.
Hey, so it is 20 marks. This one is our first assignment. So you have to be serious in our assignment, because they play a role part in a major role in awarding you or grading you or in assessment of you. Maybe I'll say so and so was good, alikuwa anapata hivi. So I want you to do that assignment. So that one will mark the end of our class, unless if there is a reaction or there is a question that someone would like to ask before we leave for the next class next week. Any reaction? Any reaction?
We are good.
Any
We are
reaction?
good, sir.
You are good. You are good.
Good job, good job.
Good job. And some of you are not talking. Here the active members is Priscilla Warimu. I can also see Nani Joan Coril. Unisawe, you are very dull in my class. I didn't expect that. But I'm very happy you have come into my class. We have learned something this evening. And now that at least when we are talking about proteins, you can say something about proteins. You are not just blank as you came. Sindio? Yes?
Yes.
Yes, thank you. Till we meet next time.
Yes. Good evening.
You too.